Hello everyone and welcome back to yet another video of the biology series. For the ones who are new here, we do NCERT and literally every line of it. And today we'll be discussing the sixth chapter, that is molecular basis of inheritance. This can serve as an audiobook as well as a last moment NCERT highlight series. So do like and watch till the end. In the previous chapter, you have learnt the inheritance patterns and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material, at least for the majority of organisms. In class 11, you have learned that nucleic acids are polymers of nucleotides. So basically, in the first paragraph, they're trying to give us an idea of the genetics that we've already done in the previous lesson. That is basically the principles of inheritance and variation. In that lesson, we see that basically on a broader scale, how did Mendel and how did the first scientific approach towards genetics arise, and how did people know that there was something that was being inherited? Say Mendel called them the factors and stuff, right? So we did that already. Now in this particular chapter, we'll go deep into it at the very molecular level, at a very basic level, जिसमें हम देखेंगे कि ये हो कैसे रहा है at the very जब हम molecule wise लेंगे तो ये कैसे हो रहा है, what molecules, what is a gene, fine, where does it lie. Deoxyribonucleic acid and ribonucleic acid are the two types of nucleic acids found in living systems. DNA acts as a genetic material in most of the organisms. We had so many experiments also proving the same. RNA, though it also acts as a genetic material in some viruses, mostly functions as a messenger. RNA has additional roles as well. It functions as adapter, structural, and in some cases as a catalytic molecule. And we know the molecules which catalyze are enzymes. And if RNA acts as an enzyme, it's known as ribozyme. In class 11, you've already learned the structures of nucleotides and the way these monomer units are linked to form nucleic acid polymers. So basically nucleic acid जो है हमारे पास ribonucleic acid है, deoxyribonucleic acid है, ये बने कहाँ से है? Monomeric units क्या है? It's the nucleotides. In this chapter, we're going to discuss the structure of DNA, its replication, the process of making RNA from DNA, जिसको transcription बोलते हैं, genetic code that determines the sequence of amino acids and proteins, the process of protein synthesis, जिसको translation बोलते हैं, जो basically हम central dogma में करेंगे, and elementary basis of their regulation. The determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. So हम genomics at the end will be talking about how do we sequence all of the, even inside the human beings, we will sequence all the genes, all the DNA material inside, fine, which is really expensive, and we'll see how it works. In the last section, the essentials of human genome sequencing and its consequences will also be discussed, जिसको हम human genomic project में करेंगे. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. Fine, आगे हम इसको देखते हैं. Let's move on to DNA now. तो सबसे पहले हमने already पढ़ा DNA ही majority में हमारे पास genetic material होता है, isn't it? We already did that. Now what is basically this DNA comprising of? ये क्या है? This is a very important lesson from NEET point of view, from board's point of view. It's really important, fine? So alright. DNA is a long polymer of deoxyribonucleotides. हमने देखा कि ribonucleotides जो है, लेकिन deoxy 2' पोजीशन पे OH नहीं होगा. डायग्रामेटिकली भी आपने बचपन से आप देखते हैं, तो वो जो होता है उसका पॉलीमर बनाएंगे, वो DNA बना देगा. The length of DNA is usually defined as number of nucleotides or a pair, क्योंकि ये pairs में होता है, ऐसा कुछ DNA helical structure जो हम आगे देखेंगे. Fine, it's just something तो हमारे पास 2 strands होते हैं, तो pairs में this pairing between the nucleotide bases, तो इनको base pairs भी बोलते हैं. तो कितने number of base pairs है या कितने nucleotides है, one and the same thing, this will give you the length of the DNA. This also is the characteristic of an organism. एक organism में, एक species में एक ही तरह का होने वाला है. For example, a bacteriophage known as 5174 has 5386 nucleotides. Very important. Bacteriophage lambda has 48502 base pairs. E. coli has 4.6 into 10 raised power 6 base pairs, and haploid content of human DNA is 3.3 into 10 raised power 9. Haploid means एक हमारे पास single set of chromosomes जहाँ पे होता है. Diploid में आप इसको you'll simply multiply it by 2 and that'll be 6.6 into 10 raised power 9 base pairs. Fine. Now let's come to structure of polynucleotide chain. Let us recapitulate the chemical structure of polynucleotide chain, DNA और RNA. ठीक, हमारे पास अब जो ये चेन बन रहा है मोनोमर यूनिट से, हम इसके स्ट्रक्चर पे आते हैं. A nucleotide has three components: a nitrogenous base, एक तो हमारे पास बेस होता है, ठीक, एक पेंटोज शुगर होता है, which can be ribose in case of RNA, it can be deoxyribose in case of DNA, and a phosphate group. There are two types of nitrogen bases. एक हमारे पास purines होते हैं और हमारे पास pyrimidines होते हैं. Fine. अब purines जो होते हैं, they can be adenine and guanine. Pyrimidines can be cytosine, uracil and thymine. साइटोसिन इज कॉमन फॉर बोथ डीएनए एंड आरएनए. थाइमीन जो होता है डीएनए में होता है, और थाइमीन की जगह आरएनए में दिस यूरासिल इज प्रेजेंट इन आरएनए एट द प्लेस ऑफ थाइमीन. A nitrogenous base is linked to the OH of the first carbon of the pentose sugar through a N-glycosidic linkage to form a nucleoside. Basically, हम सबसे पहले क्या लेंगे, हमारा base लेंगे. Fine. We'll first create actually the DNA molecule. Say we'll take a base, we'll take a nitrogenous base. It can be a purine, it can be a pyrimidine. इसको हमने link किया OH hydroxyl group किसका, pentose sugar. Pentose sugar जो है उसमें hydroxyl group होगा. उसके साथ जब हमने, अब माने आपका pentose आ गया और आपका nitrogenous base आ गया, we'll call nucleoside. एंड ये किससे जुड़ेगा, ग्लाइकोसिडिक लिंकेज से जुड़ेगा, सच एज एडेनोसिन और डीऑक्सीएडेनोसिन. फाइन. देन वी हैव guanosine और deoxyguanosine, सिटिडीन, deoxycytidine, यूरिडीन और deoxythymidine. फाइन. क्योंकि यूरिडीन के नॉट एग्जिस्ट बिकॉज इन RNA we have uracil, in place of that in DNA we have thymine. Fine. When a phosphate group, अब तीन चीजें, दो हमने जोड़ ली. The third thing is phosphate group is linked to the OH of 5' carbon of a nucleoside. हाँ, 5' कार्बन पे अब किसका OH आएगा, फॉस्फेट ग्रुप का आएगा. फाइन. अब इसको लिंक करेंगे थ्रू फॉस्फोएस्टर लिंकेज. A corresponding nucleotide or deoxynucleotide depending upon the type of sugar present is formed. Two nucleotides are linked through, अब दो न्यूक्लियोटाइड्स को आपस में जोड़ना है क्योंकि हमें पॉलीमर चेन बनाना है. 3' 5' phosphodiester linkage जो है to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has, अब क्या किया, 3' 5' पे फॉस्फोडाइस्टर बॉन्ड लगा लिया. फाइन. पहले तो हमने 5' कार्बन पे फॉस्फेट का OH लगाया. 3' पे जो हमारा पहला कार्बन था पेंटोज शुगर का, उसके साथ हमने किसको जोड़ा, पहले तो नाइट्रोजन बेस को एन ग्लाइकोसिडिक बॉन्ड से. नाउ व्हाट वी हैव नेक्स्ट इज हियर. ऑलराइट. एंड a polymer thus formed has at one end a free phosphate moiety at 5' end of sugar, which is referred to as 5' end of polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH of 3' group, ठीक, carbon group, which is referred to as the 3' end. तो 3' end पे हमारे पास OH होगा, हाइड्रोक्सिल होगा, 5' पे हमारे पास होगा phosphate group. The backbone of a polynucleotide chain is formed due to a sugar and the phosphate. तो sugar और phosphate से backbone बनाएंगे. Nitrogenous bases जो है they will project from the backbone. ठीक है, जैसे हमारी body में the backbone, from it will project the ribs. Fine. The nitrogenous base is linked to sugar moiety project from the backbone. In RNA, every nucleotide residue has an additional OH group present at 2' position in the ribose. तो क्योंकि हमने deoxy में ये हटा लिया होता है, लेकिन RNA में ये 2' position पे होता है. Also in RNA, the uracil is found in the place of thymine. What is basically thymine? It's nothing. You have methylated the uracil, you've made it more stable. ठीक है, तो उसको बोलते हैं 5-methyl uracil, which is another chemical name for thymine. DNA as an acidic substance present in nucleus, deoxyribonucleic acid होता है. It was first identified by Frederick Mischer in 1869. He named it as nuclein. So nuclein term was used by Mischer first. Fine. So however, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. But सबसे पहले, the presence of an acidic substance inside the nucleus, it was first given by Mischer, and he called it nuclein. Fine. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. तो सबसे पहले जो structure दिया, although it was very simple, we had eventually हमारे पास Hershey Chase का ही experiment था जो हम अभी तक मानते हैं, but then Watson and Crick model was also, one of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chain. So they gave a very important aspect of the base pairing thing. ठीक है, कि जो हमारा बैकबोन है वो जिसका भी बना है दिस बेस पेयरिंग बिटवीन द टू strands of the polynucleotide chain, and this was given by the double helical structure only. However, this proposition was also based on the observation of Chergaff that for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equals to 1. ये उसने बोला था बेसिकली जितने अगर पेयरिंग हो रही है इसका मतलब जितने एडेनिन होंगे उतने थाइमीन होंगे और जितने ग्वानिन होंगे उतने साइटोसिन होंगे, बिकॉज वी हैव AT डबल बॉन्ड, वी हैव GC ट्रिपल बॉन्ड. एडेनिन थाइमीन के साथ पेयर कर रहा है, यू नीड टू हैव कॉम्प्लीमेंटेशन ओवर देयर. कॉम्प्लीमेंट्री स्ट्रैंड्स आर गोइंग टू bind together to form a double helical structure. Fine. Moving on. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore if the sequence of bases in one strand is known, then the sequence in other strand can be predicted. अगर आपको एक strand का पता है दूसरे का आप predict कर सकते हैं क्योंकि वो complementary होते हैं. Also, if each strand from a DNA, let us call it as a parental DNA, act as a template for synthesis of a new strand. ठीक है, अगर एक को हमने माना template, एक DNA से ही दूसरा बनेगा, fine. अगर दो हमारा double stranded DNA उसके एक strand को ले लिया, उससे हमने नया बनाया, उसको template बोलेंगे. The two double stranded DNA जिनको जो daughter DNA उससे बनेंगे, they would be identical to the parent DNA molecule. Because of this, the genetic implications of the structure of DNA became very clear. Fine. अब हम देखते हैं डबल हेलिकल स्ट्रक्चर क्या बोला अकॉर्डिंग टू वाटसन एंड क्रिक मॉडल. इट इज मेड ऑफ टू पॉलीन्यूक्लियोटाइड चेन्स. वी आर डिस्कसिंग डीएनए अकॉर्डिंग टू वाटसन एंड क्रिक मॉडल. पॉलीन्यूक्लियोटाइड चेन्स होंगे where the backbone is constituted by sugar phosphate and the bases would project inside. The two chains have anti-parallel polarity. इसका मतलब क्योंकि complementation आना चाहिए, you know, if one chain has the polarity 5' to 3', the other has 3' to 5'. The bases in two strands are paired through hydrogen bonds forming base pairs. Adenine forms two hydrogen bonds with thymine from the opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds, like I already said. As a result, always a purine comes opposite to a pyrimidine. ठीक है, हमेशा एक purine pyrimidine के opposite आएगा. This generates approximately uniform distance between the two strands of the helix. Fine, the two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer. Fine, and there are roughly 10 base pairs in each turn. ठीक है, consequently the distance between a base pair and a helix is approximately 0.34 nanometer. Just numerical values that you have to remember actually. ठीक है, ये यहाँ पे इन्हें दिया हुआ है the hydrogen bonds. ठीक है, G का C के साथ, then G, C, A, T, 2 double bonds only. Here you have 3 of them. Fine, so this is basically the double stranded chain. These are your base pairs again, complementary to the other. Fine, so adenine, thymine, guanine, cytosine. Fine. The plane of one base pair stacks over the other in the double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. तो एक के ऊपर एक base pairs will be stacked. Compare the structure of purines and pyrimidines. Can you find out why the distance between two polynucleotide chains in DNA remains almost constant? दो chains अगर आपस में wind भी हो रहे हैं right-handed fashion में भी, yet उन दोनों के बीच का distance same रहेगा. क्यों? क्योंकि हमारे पास एडेनिन हमेशा प्यूरिन और पिरिमिडीन ही ऑपोजिट में आएंगे, तो उनके बॉन्ड्स मोर और लेस सेम लेंथ के होंगे. Alright, the proposition of a double helix structure for DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA to protein. This is the simplest form of the central dogma that Francis Crick gave. He said that DNA will get transcribed into RNA, it will get translated into proteins, and that's how life would work. This was the central dogma. ये अगर DNA आपस में खुद ही ये कर रहा है, it's replication, then transcribe करके mRNA दिया है, but mainly RNA को ही pertain कर रहा है, फिर translate करके proteins बनेंगे. We'll be talking about in depth all of these terms. Fine. It's just the basics. In some viruses, the flow of information is in reverse direction, that is from RNA to DNA. अच्छा, retroviruses होते हैं जिनमें RNA से DNA बनता है, उनके पास reverse transcriptase होता है. Fine, that's an enzyme present in them, which we'll also be discussing later on. Moving on, we have packaging of the DNA helix. अब यहाँ packaging करने का यही क्यों आया? Why did it in the first place come? Why do we need to package the DNA? अभी तो ठीक है ना, helical structure ठीक है, double stranded है, it's fine. We need to pack this because उसका length बहुत ज़्यादा है और हमें वो सारे को accommodate करना है अपने body के अंदर, अपने cells के अंदर. Fine. So you need to do compaction. That is what you mean by packaging of the DNA helix. taken the distance between two consecutive base pairs as 0.34 nanometer, if the length of DNA double helix in a typical mammalian cell is calculated, ठीक है, जितने number of base pairs है with the distance between two consecutive base pairs, it comes out to be approximately 2.2 meters, हाँ, mammals में. A length that is far greater than the dimension of a typical nucleus. ठीक है, nucleus का तो minus में चला जाता है, in the order of -6 they have given. ठीक, तो कैसे हम meters का coil इसमें बंद करा सकते हैं? Fine. In prokaryotes such as E. coli, they do not have a defined nucleus. The DNA is not scattered throughout the cell. Fine. DNA being negatively charged is held with some proteins that have a positive charge in a region termed as the nucleoid. तो prokaryotes में क्या होता है? Nucleus तो नहीं है, लेकिन DNA scattered नहीं होता है. It's present in a certain place. जहाँ पे इसको proteins जो है positive charge वाले, वो nucleoid region में इसको bind करेंगे. DNA is negatively charged. The DNA nucleoid is organized in large loops held by proteins. अब eukaryotes में ऐसा नहीं होता. Organization is much more complex. There's a set of positively charged basic proteins called histones. Histones का basic nature है, इस पे भी नीट में सवाल आ चुका है. फाइन. एंड दिस इज सेट ऑफ इट्स ऑब्वियसली पॉजिटिवली चार्ज. अ प्रोटीन acquires charge depending upon the abundance of amino acids residues with charge side chains. Obviously जिस amino acid का वो बना होगा उसमें charge होना चाहिए. Histones are rich in the basic amino acid residues lysine and arginine. Lysine, arginine are basic amino acids जो histones को basic nature confer करते हैं. Fine. Both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. ठीक है, histone किस में convert हो जाएगा? Histone octamer में. ठीक. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called a nucleosome. अब क्या है, हमारे पास पॉजिटिवली चार्ज हिस्टोन ऑक्टामर आ गया, उसके इर्द गिर्द किसने खुद को रैप किया? डीएनए ने, जो था नेगेटिवली चार्ज, और क्या बनाया? न्यूक्लियोसोम बनाया. अ टिपिकल न्यूक्लियोसोम कंटेन्स 200 बेस पेयर्स ऑफ डीएनए हेलिक्स. अगेन वेरी इंपॉर्टेंट. हाँ, एक न्यूक्लियोसोम में 200 बेस पेयर्स होंगे. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin. Basically, chromatin जो होता है वो क्या होता है? Basically, repeated nucleosomes is chromatin. Thread-like stain-colored bodies seen in nucleus. The nucleosomes in chromatin are seen as beads on this string structure. ठीक है, अगर आप इसको इलेक्ट्रॉन माइक्रोस्कोप के अंदर देखेंगे, see, these are like beads on the string. यही हमारा chromatin होता है. Basically क्या होता है? Nucleosomes होते हैं. Fine. Next we have theoretically, how many such beads do you imagine are present? Yeah, the beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of the cell division to form. ये हम already कर चुके हैं cell cycle में कि क्या होता है? metaphase stage पे ये further condense हो जाता है, further coiling will take place. Fine. So the packaging of chromatin at higher level requires additional set of proteins that are collectively referred to as non-histone chromosomal proteins. ये further package करने में इसको help करते हैं. हम कुछ नहीं कर रहे, we're just doing the compaction of the whatever DNA we have. In a typical nucleus, some region of chromatin are loosely packed, और वो stain भी light करता है, और उसको हम euchromatin बोलते हैं. This chromatin basically, this is the ट्रांसक्रिप्शनली एक्टिव पार्ट जो एक्चुअली ट्रांसक्राइब कर सकता है, यूक्रोमेटिन. लेकिन हमारे पास कहीं जगहों पे क्रोमेटिन बहुत डेंसली पैक्ड होता है एंड इट स्टेन्स डार्क, उसको हेटेरोक्रोमेटिन बोलते हैं. ठीक है, विच इज नॉट ट्रांसक्रिप्शनली एक्टिव. द सर्च फॉर जेनेटिक मटीरियल. Even though the discovery of nuclein by Mischer and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. ठीक है, अभी पता नहीं था कि DNA ही है या नहीं. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Mendel, Sutton, Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. ऐसी कौन सी चीज़ थी जो ट्रांसफर हो रही थी, जो इनहेरिट कर रहे थे, वो अभी भी पता नहीं था. तो अभी भी साइंटिस्ट इसी तलाश में कि whether it's DNA or it's something else. So we have Griffith's transforming principle, very important. This has also been asked कि transformation किसने दिया? It was Griffith. In a series of experiments with Streptococcus pneumoniae, which is the bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism had changed in physical form. Basically क्या हो रहा था? एक स्ट्रेन ले लिया निमोनिया, स्ट्रेप्टोकोकस निमोनिया का, उसको एस स्ट्रेन बोला. एस स्ट्रेन जो था वो जो था इन्फेक्शियस था, वो वायरलेंट था, उससे मर रहे थे माइस. ठीक. दूसरा ले लिया आर स्ट्रेन. आर स्ट्रेन जो था उससे माइस बच रहे थे, मतलब वो वायरलेंट नहीं था. एंड सी व्हाट ही कुड कंक्लूड फ्रॉम दिस एक्सपेरिमेंट. When pneumococcus bacteria are grown on a culture plate, some produce smooth, shiny colonies, S strain, while others produce rough colonies. This is because the S strain bacteria have a mucus or a polysaccharide coat, while R strain does not. Mice infected with the S strain, जो virulent strain है, they die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. ठीक. अब S strain का क्या है? इंजेक्ट कर लिया तो माइस विल डाई, आर स्ट्रेन माइस विल लिव. ठीक. Griffith was able to kill bacteria by heating them. He observed that heat killed S strain bacteria injected into mice did not kill them. Obviously क्योंकि S strain को जब हमने heat kill किया, अब उनमें कुछ वैसी चीज रही नहीं. We injected it into mice, the mice would live. अगर हमने heat killed S strain और R strain ले लिया, R strain को live ले लिया, when you injected it into mice, the mice died. लेकिन हमने तो हीट किल्ड एस्ट्रेन लिया था और पिछले एक्सपेरिमेंट के अकॉर्डिंग इसने तो कुछ नहीं करना था माइस को. दिस इज व्हाट कंफ्यूज्ड हिम. सी व्हाट ही डिड. He concluded that the R strain bacteria had somehow been transformed. R strain में तो ऐसा कुछ नहीं था कि वो virulent होता है या वैसा कुछ, but वो transform हो चुका था. किससे? By the heat killed S strain bacteria जो हमने उसके साथ डाले थे. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. कुछ S strain में ऐसी चीज थी जो R strain ने ले ली, transform हो गया और अपने इर्द गिर्द वही polysaccharide coat बनाया जो S strain में था और ये भी virulent बन गया. This must be due to the transfer of the genetic material. However, this biochemical nature of genetic material was not defined from his experiments. He still couldn't explain. He just explained that there was something that was being transformed, but he couldn't say what was the material basically. अब biochemical characterization ये किसने दिया था? Again, very important. Avery, MacLeod, McCarthy. These three names you have to remember. They were the ones who worked towards the biochemical principle of transformation. Fine. The genetic material was thought to be a proton. इनसे पहले, prior to the work of these people, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals, proteins, DNA, RNA, etc. from the heat-killed S cells to see which ones could transform live R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria to be transformed. What did they do basically? उनने biochemicals निकाले, DNA ले लिया, protein, RNA, ठीक है, heat killed S strain, and they had to see what was the chemical that was getting transformed into the live R cells. Fine. And they discovered that it was DNA alone that was responsible for the S bacteria, for the R bacteria to become virulent. Fine. They also discovered that protein digesting enzymes, proteases, RNA digesting enzymes, RNAs did not affect transformation. So the transforming substance was not a protein or RNA. जब उन्हें इनके lysis कराया तो तब तो ट्रांसफॉर्मेशन पे कोई फर्क नहीं पड़ा. बट डाइजेशन में डीएनएस डिड इनहिबिट ट्रांसफॉर्मेशन. जब डीएनएस था, डीएनए नहीं रहेगा. जब डीएनए नहीं रहेगा, ट्रांसफॉर्मेशन भी नहीं होगा. सजेस्टिंग दैट द डीएनए कॉज द ट्रांसफॉर्मेशन. दे कंक्लूडेड दैट डीएनए इज द हेरिडिटरी मटेरियल, बट नॉट ऑल बायोलॉजिस्ट्स वर कन्विंस्ड. तो अभी भी कन्विंस नहीं हुए थे. Now, the genetic material is DNA. The unique vocal proof that DNA is a genetic material came from the experiments of Hershey and Chase. ठीक, ये एक्सपेरिमेंट आपको याद रखना है. They worked with viruses that infect bacteria called bacteriophages. उनने bacteriophages पे काम किया. The bacteriophage attached the bacteria and its genetic material that enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. तो बैक्टीरिया में क्या होता है? जो बैक्टीरियोफेज घुसते हैं, जो वायरसेस घुसते हैं, तो बैक्टीरिया के मैकेनिज्म को पता नहीं चलता कि ये हमारे अपने सेल्स हैं जिनको हम बना रहे हैं या फिर ये वायरस सेल्स हैं. तो वो इवेंचुअली क्या करते हैं? Viral genetic material को अपना समझ के they will produce more viral particles. Fine. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. ऐसी कौन सी चीज थी जो बैक्टीरिया को एंटर कर रही थी? Because eventually वही genetic material था. Fine. Whether it was the protein or it was the DNA. अभी यही हमारा question था. They grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. अब आपको याद रखना है radioactive phosphorus हमारा DNA में present होता है, हमने अभी देखा, और sulfur जो है प्रोटीन में present होता है. Fine. Viruses grown in the presence of radioactive phosphorus contain radioactive DNA. We know this, but not radioactive protein because DNA contains phosphorus but protein does not. Fine. Similarly, इनका बहुत, this is a huge contribution. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA because DNA does not contain sulfur. ठीक, phosphorus ले लिया, sulfur ले लिया. कौन से ले लिया? radioactive isotopes ले लिया. phosphorus DNA में होता है, sulfur protein में होता है. Moving on. Radioactive phages were allowed to attach to E. coli bacteria. Then as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. हम अलग कर रहे हैं अब इनको because already they have attached to the bacteria. The bacteriophage is attached to the bacteria. Bacteria which was infected with virus that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. अब infected bacteria में radioactive DNA था, यानी वो रेडियोएक्टिव थे. इसका मतलब वही, अब जो रेडियोएक्टिव होगा वही हमारा इन्फेक्टेड होगा. इसका मतलब ऐसी कौन सी सब्सटेंस जो ट्रांसफर हुई थी? इट वाज डीएनए. फाइन. बैक्टीरिया दैट वर इन्फेक्टेड विद वायरसेस दैट हैड रेडियोएक्टिव प्रोटीन्स वर नॉट रेडियोएक्टिव. रेडियोएक्टिव प्रोटीन्स में तो डाल दिया बैक्टीरिया को, बट हमारा बैक्टीरिया रेडियोएक्टिव नहीं बना बिकॉज प्रोटीन बैक्टीरिया में नहीं जा रहा था. दिस इंडिकेट्स दैट प्रोटीन डिड नॉट एंटर द बैक्टीरिया फ्रॉम द वायरसेस. डीएनए इज देयरफॉर द जेनेटिक मटेरियल दैट इज पास्ड फ्रॉम वायरस टू द बैक्टीरिया. Fine. So first you infected them, you blended them in, and then centrifugation say you had to separate them. Fine. So this is the P32 detected cells. There is no radioactivity in the supernatant क्योंकि वो सारा सेल के अंदर चला गया. Fine. यहाँ पे radioactive supernatant होगा यानी सल्फर है. सल्फर है radioactive substance क्योंकि वो सेल के अंदर नहीं जाएगा, वो प्रोटीन है. Properties of genetic material, DNA versus RNA. From the foregoing discussion, it is clear that the debate between proteins versus DNA as the genetic material was unequivocally resolved from Hershey-Chase experiment. It became an established fact that it is DNA that acts as genetic material. However, it is subsequently became clear that in some viruses, RNA is the genetic material. अब कभी कभी हमने देखा हर दफा DNA is not the genetic material. Sometimes RNA can also act as the genetic material. For example, TMV. They've given tobacco mosaic, virus, QB bacteriophage, etc. Now again, these examples are important. Answer to some of the questions such as why DNA is the predominant genetic material whereas RNA performs dynamic functions of messenger and adapter has to be found from the differences between chemical structures of the two nucleic acid molecules. Now it says there are differences between DNA and RNA. There's a certain reason that DNA is the genetic material, RNA is not. Fine. Though RNA हम देखते हैं it's more reactive, it acts as a catalyst, it acts as a messenger, it's more reactive even, so it, and even transmission is very easy with the help of RNA. But what makes DNA more stable in terms of DNA being the genetic material in most of the cases, that will be dealt with subsequently. Fine. Moving on, a molecule that acts as a genetic material. अब ऐसे कैसे properties होने चाहिए हमारे genetic material में जो कि possible है कि फिर वो एक genetic material की तरह act करे. What are the properties it must fulfill? It should be able to generate its replica. Replicate करना पड़ेगा. It should be stable chemically and structurally. It should provide the scope for slow changes. Mutation आना चाहिए जो evolve करेगा हमें. Fine. Mutation होना चाहिए, replication होना चाहिए. There should be stability. It should be able to express itself in the form of Mendelian characters. It should be transmitted, inherited, it can express. If one examines each requirement one by one, because of rule of base pairing and complementarity, both the nucleic acids have the ability to direct their duplications. ठीक, दोनों duplicates बना सकते हैं. The other molecules in the living system such as proteins fail to fulfill the first criteria itself. पहला ही है कि protein अपने आप का replica नहीं बना सकता. Fine. The genetic material should be stable enough not to change with different stages of life cycle, age or with change in physiology of the organism. Stability बहुत important है. इसको वैसे ही रहना है no matter what the organism body, the rest of the body is going through. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that heat which killed the bacteria at least did not destroy some of the properties of genetic material. हम heat killed strain ले रहे थे लेकिन फिर भी transformation हो रहा था S strain से into the R strain. This was only possible that because of this genetic stability of the DNA. This now can easily be explained in light of the DNA that the two strands being complementary if separated by heating come together. क्योंकि complementarity है, अगर आप उनको अलग भी करेंगे उनका फिर they have a tendency to join back when appropriate conditions are provided. Further, 2-OH group present at every nucleotide in RNA is a reactive group. 2- पे जो OH group है वो reactive group है, RNA को labile बनाएगा, easily degradable. It's also known to be a catalyst. It's very reactive. Hence, DNA is chemically less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, DNA is a better genetic material. RNA के पास एक additional hydroxyl group है, वो उसको reactivity बढ़ाता है, ठीक है? लेकिन DNA के पास वो नहीं है. It's deoxyribonucleic acid. As a result, it's a better genetic material. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Detailed discussion about this requires understanding the process of repair in DNA, and you'll study these processes in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutates at a faster rate. क्योंकि वो unstable है, तो वो mutation भी जल्दी करेगा because it's really reactive as well. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. ऐसे viruses जिनमें RNA acts as a genetic material जल्दी mutate करते हैं और evolve भी बहुत जल्दी होते हैं, ठीक, because वो unstable होता है, वो बहुत reactive होता है. RNA can directly code for the synthesis of proteins, hence can easily express the characters. क्योंकि RNA के बाद सीधा translation होता है और proteins बनते हैं. DNA को however is dependent on the RNA for the synthesis of proteins, ठीक है, क्योंकि जो protein synthesis machinery है, it is evolved around the RNA. तो ये कह रहा है बिकॉज हमने सेंट्रल डॉगमा में पहले ही देखा था. पहले तो हमारे पास आता है you have transcription and then you have translation. So DNA is dependent upon the RNA for the synthesis of proteins. RNA का ऐसा कोई मसला नहीं. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. Transmission के लिए RNA बेहतर है, मगर stable ज़्यादा storage के लिए it is DNA. Fine. Moving on to now RNA. अब DNA तो हम कर ही चुके हैं. The differences even we are done with. Moving on to RNA. From the foregoing discussion, immediate question becomes evident, which is the first genetic material. यानी पहले कौन आया, फिर बाद में हम मानते हैं advancement आया या evolve होके कुछ और आया. But what was it which came first? RNA was the first genetic material. Fine. To begin with, there's now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as a catalyst. Fine. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA only with chemical modifications that make it more stable. DNA being double-stranded and having complementary strand further resist changes by evolving a process of repair. Fine. Changes नहीं होंगी, reactivity कम होगी, stability ज़्यादा होगी in case of DNA. Why? Because it is, of course, it has evolved from RNA and it's basically an advancement. Fine. Originally we had RNA as the genetic material, but since it was a catalyst, it was reactive, we had to find a stable alternative. Fine. And that was our DNA molecule. And we have so many more differences that we already did, which will confer extra stability to the DNA molecules. So for example, not having the OH additional group, fine, etc, etc. But however, you'll remember that transmission ke liye it's only RNA that is, that can act as a faster transmitter. Now, what is replication? While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This is what Watson and Crick said. उन्होंने बोला, हमें समझ आ गई है भाई, दो strands है, और वो क्या है उनके बीच में दिस बेस पेयरिंग, यानी देयर इज अ कॉपिंग मैकेनिज्म जिसको हम आगे पढ़ने वाले. द स्कीम सजेस्टेड दैट द टू स्ट्रैंड्स वुड सेपरेट एंड एक्ट एज अ टेम्पलेट फॉर द सिंथेसिस ऑफ न्यू कॉम्प्लीमेंट्री स्ट्रैंड्स. फाइन. इफ दिस डबल हेलिक्स वुड सेपरेट, वी हैव नाउ टू सिंगल स्ट्रैंडेड DNA molecules. Each can act as a template and give rise to DNA, daughter DNA. Fine. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. एक तो parental अपना है और जिसका complementary, now another daughter strand will come. Fine. That will come afterwards. So that is why this scheme is known as the semi-conservative DNA. Again, very important. Semi-conservative क्यों? क्योंकि एक strand parental होगा जो template DNA से आया है और दूसरा daughter DNA strand होगा which is the new strand which is formulated complementary to the template strand. This is the semi-conservative nature of the DNA replication. The experimental proof now. It is now proven that DNA replicates semi-conservatively. It was shown first in E. coli and subsequently in higher plants, organisms such as plants and human cells. Messelson and Stahl performed the following experiment. Again, किसने हमें experimentally prove किया कि DNA replication is semi-conservative in nature? It was Messelson and Stahl. Again, very two important names. ठीक? They grew E. coli in a medium containing ammonium chloride, इसका heavy isotope लिया nitrogen का as the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation as cesium chloride density gradient. Now it says that N15 is not a radioactive isotope. It can be separated from N14 only on the basis of densities. ये दोनों isotopes है, दोनों radioactive नहीं है, तो हम इनको सिर्फ cesium chloride का एक density graded medium सा ले लेंगे, ठीक है. उसमें जब centrifugation करेंगे ना, different depths पे हमें different molecules मिलेंगे, and you can actually understand density के basis पे हम उनको separate कर सकते हैं. N15 heavy है, N14 normal वाला है जो light होने वाला है, fine. Then they transferred from the cells into a medium with normal ammonium chloride, जिसमें N14 ले लिया. N14 आप समझ रहे हैं जो normal हमारा nitrogen है, and took samples at various definite time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. The various samples were separated independently on cesium chloride gradients to measure the densities of DNA. Now what did they infer from here? Say you have this heavy DNA. ये हमारा cesium chloride gradient है, जो भी you have only heavy isotopes. Then you have हमने इनको क्या किया afterwards? You're trying, this is basically separation of DNA by centrifugation. Let's read it first out. Thus, the DNA that was extracted from the culture 1 generation after the transfer from N15 to N14 medium, 20 minutes के बाद हमने N15 से किस medium में डाला? N14 में. Fine. They're a hybrid of intermediate density. क्योंकि पहले हमने N15 वाले density ले लिया, उसके बाद हमने N14 वाला ले लिया. Fine. Now intermediate density मिला. DNA extracted from the culture after another generation. अब 40 minutes के बाद दूसरे में डाला, was composed of equal amounts of this hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, फिर इसके बाद एक और cyclic process would take place. Very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes was performed on Vischer Faba by Taylor and colleagues. The experiments proved that the DNA in chromosomes also replicates semi-conservatively. ठीक है, तो बाद में Vischer Faba पे Taylor did the similar experiment, ठीक, and he proved that DNA in the chromosome will replicate semi-conservatively. यानी parental template का जो already असर होगा, that will still remain there. Fine. Even if you're changing the media, yet जो हमारा basic था, intermediate hybrid जो था, हमारे पास hybrid DNA जो मिला था N15 और N14 वाला, you'll still find that because the DNA would replicate semi-conservatively. The machinery and the enzymes. In living cells such as E. coli, the process of replication requires a set of catalyst enzymes. The main enzyme is referred to as DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. So again ये यही कह रहा है कि क्योंकि हमें एक DNA template से हमें इसका रेप्लिकास बनाने हैं, तो हमारे पास एक ऐसा एंजाइम होना चाहिए जो उस प्रोसेस को कैटलाइज करे. ठीक, सो वी हैव अ डीएनए डिपेंडेंट डीएनए पॉलीमरेज. डीएनए पॉलीमरेज मतलब हम डीएनए के ही पॉलीमर्स बना रहे हैं, हम डीएनए को ही रेप्लिकेट करा रहे हैं. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. So efficiency level भी इनका बहुत high होना चाहिए. E. coli that is only 4.6 into 10 raised power 6 base pairs. Compare it with human whose diploid content is 6.6 into 10 raised power 9 base pairs. Completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pairs per second. तो अगर हम on an average देखें, it is 2000 base pairs per second. That is बहुत ज़्यादा efficient enzyme है. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Genetic material है, there is no margin of making a risk. Fine. Any mistake during replication would result into mutations. Furthermore, energetically replication is very expensive. बहुत energy लगता है. Deoxyribonucleoside triphosphates serve dual purposes. ठीक है, जो हमारे triphosphates, so in addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphates in a deoxynucleoside triphosphate are high energy phosphates, same as in case of ATP. So they'll also, besides acting as substrates, जिसका वो लोग polymerase करेंगे, जिसका वो लोग polymer बनाएंगे, उसके अलावा this is also going to act as a source of energy because our DNA replication, the machinery, this requires a large amount of energy, just like ATP would do. ठीक है, because बिकॉज है तो ये भी ट्राइफॉस्फेट्स, राइट? इन एडिशन टू डीएनए डिपेंडेंट डीएनए पॉलीमरेज, मेनी एडिशनल एंजाइम्स आर रिक्वायर्ड टू कंप्लीट द प्रोसेस ऑफ रेप्लिकेशन विद हाई डिग्री ऑफ accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occur within a small opening of the DNA helix, referred to as replication fork. हम पूरा DNA को पूरे को unwind नहीं कर सकते. We cannot separate the two strands in its entire length. So what we do is there's a certain region जिसको हम replication fork बोलते हैं, Y-shaped, region, जिसमें this replication is going to take place. ये unwound helix का जब आप tension आ जाता है वहाँ पे, you also have topoisomerase for that, for releasing that tension when you're unwinding the two strings of the double stranded DNA, तो एक Y shaped structure बन जाता है. Fine, so that's like this. ठीक, कुछ ऐसा. और यही हमारा रेप्लिकेशन फोर्क होता है और इस रीजन में हमारा रेप्लिकेशन होता है. यहाँ से तो कंटिन्यूअसली यू हैव द टू स्ट्रैंड्स. The DNA-dependent DNA polymerase catalyze polymerization only in one direction, that is 5' to 3'. This creates some additional complications of the replicating fork. Consequently, on one strand, the template with polarity 3' to 5', the replication is continuous. जो 3' 5' वाला होगा उसमें replication constant होगी. क्यों? Continuous क्यों होगी? क्योंकि इसमें 5' 3' में करेगा DNA-dependent DNA polymerase, polymerization 5' 3' करेगा. और ये अगर template है, इसका complementary कौन होगा? 5' 3'. Fine, that is continuous. While on the other hand, the template with polarity 5' to 3', उसमें कौन सा बनेगा हमारा डॉटर DNA? It will be 3' to 5'. लेकिन 3' 5' continuous नहीं होता, it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase, and those fragments are known as Okazaki fragments. The DNA polymerases on their own cannot initiate the process of replication. ठीक है, खुद से initiation नहीं हो सकता. Is it clear? You need to have something else. Also, the replication does not initiate randomly at any place in DNA. There's a definite region in E. coli DNA where the replication originates. ऐसे regions को हम ORI sites, which we did already, biotech में किया था. Such regions are termed as origin of replication. ऐसे sites जहाँ पे replication starts. Self-explanatory term. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. ठीक है, क्यों चाहिए होता है वेक्टर? पहले जब भी हम recombinant DNA, I told you we've done this in biotech only. क्या होता है कि जो vectors होते हैं वो देते क्या है? Basically they provide an origin of replication. Fine, that's why we need a vector whenever we go for genetic recombination. Moving on, we have further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. Very important. S phase of the cell cycle में synthesis phase में DNA replication starts. The replication of DNA in cell division cycle should be highly coordinated. Cell division और DNA replication बहुत proper timing पे होना चाहिए. अगर cell division नहीं हुआ, that's what it's trying to say. A failure in cell division after DNA replication results into polyploidy, which is a chromosomal anomaly. Polyploidy यानी बहुत सारे sets of chromosomes रहेंगे क्योंकि DNA has not, although DNA is replicated, cells divide नहीं हुई. And this is lethal. In plants में it can be useful, but in case of animals, it's highly lethal. ठीक है? Transcription पे आते हैं हम लोग अब. This is something given over here, replicating fork. I told you it's Y-shaped. So अगर ये हमारा template DNA है 3' to 5', fine. इसमें जब बनेगा तो वो बनेगा हमारा, see if this is like this. अगर यहाँ से जा रहा है 5' to 3', तो इसको वहाँ से आना पड़ेगा 5' to 3' और ये fragments में बनेगा. इनको हम Okazaki fragments बोलते हैं. वहीं अगर हमारे पास 3' to 5' एक हमारे पास continuous strand है, तो इसका जो 5' 3' बनेगा, even that will be continuous, no? Because 5' 3' DNA dependent DNA polymerase इसी direction में बनाता है. This is continuous synthesis. Fine. Now transcription. Moving on to the first part of the central dogma. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Fine. एक strand ले लिया DNA का, उससे information RNA में डालेंगे, इसको transcription कहते हैं. Here also the principle of complementarity governs the process of transcription, except the adenosine complements now forms base pair with uracil instead of thymine. पहले हम AT bonds देखते थे, अब हम क्या देखेंगे? Basically thymine की जगह RNA में uracil आता है, right? So this will be the difference. बाकी सारा complementarity will be followed. However, unlike in the process of replication, which once set in, the total DNA of an organism gets duplicated. In transcription, only a segment of DNA and only one of the strands is copied into RNA. ठीक है? RNA single stranded ले लिया हमने, और ये जो RNA है ये basically is just a segment of the DNA that we are transcribing. Fine. So it's different from DNA replication जो हम अभी तक कर रहे थे. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Fine. So only a certain region of the DNA would be transcribed over here. Why both the strands are not copied during transcription has the simple answer. क्यों? अगर दोनों strands का हमने बना लिया, fine, दोनों का RNA बने, उनमें again complementarity होगी, वो again जुड़ जाएंगे. It'll again be double stranded. First, if both strands act as template, they would code for RNA molecule with different sequences. ठीक है? And in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. अब ये बोल रहा है basically दो strands है तो उससे दो different sequences बनेंगे. Fine? और अगर protein के लिए code करेंगे तो amino acids का जो the sequence of amino acids it would also be different. Hence, one segment of the DNA would be coding for two different proteins. क्योंकि एक DNA से दो chains आ रहे हैं, दो templates आ रहे हैं, यानी एक ही DNA segment जो है दो different proteins के लिए code करे, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules if produced simultaneously would be complementary to each other, hence would form a double-stranded RNA. ये हमने already get. This would prevent RNA from being translated into protein and the exercise of transcription would become a futile one. तो फिर हम कर ही क्यों रहे हैं transcription अगर हमें वापस DNA double-stranded RNA मिल रहा है जो बाद में it's not capable of translating into proteins. Fine. Moving on now, we have transcription unit. A transcription unit in DNA is defined primarily by three regions in the transcription unit. यानी कौन सा वो unit है DNA का जिसको हम transcribe करके RNA बनाते हैं. mRNA book ने दिया है, but it's RNA in a broader sense. So you need to have a promoter region, the structural gene, and a terminator. Alright. There's a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA-dependent RNA polymerase, अब हम क्या करें? DNA-dependent RNA polymerase ले रहे हैं. Fine, क्योंकि है तो DNA पे dependent, बन तो DNA से रहा है, लेकिन बन क्या रहा है? RNA बन रहा है. This is transcription, right? Also catalyze the polymerization in only one direction, that is 5' to 3'. DNA dependent DNA polymerase भी 5' to 3'. फिर हमने देखा था कैसे Okazaki fragments आते हैं, you had to join them by DNA ligase. The strand that has the polarity 3' to 5' acts as a template and also referred to as the template strand. अब जिसका 3' 5' होगा एक ही strand लेना है, वो that will act as a template because 5' to 3' में continuously हमारा जो है उसका RNA transcribe होता रहेगा. The other strand now which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil, है ना? है तो वो DNA का ही strand, लेकिन वो किसको resemble करेगा? RNA को, क्योंकि RNA जो है वो दूसरे strand की complement बना है, दूसरे strand की complement ही वो पहला strand था. If you're able to create a picture in your mind, is displaced during transcription. Strangely, this strand, which does not code for anything, is referred to as coding strand. हालांकि ये जो हमारा 5' 3' end है वो किसी भी चीज़ के लिए कोड नहीं कर रहा. क्यों? क्योंकि 3' 5' is the template और उससे 5' 3' हमारा RNA बनेगा. लेकिन अब जो 5' 3' उसको कोडिंग स्ट्रैंड क्यों बोलते हैं? क्योंकि उसका सीक्वेंस और हमारा जो RNA बना उसका सीक्वेंस सेम है. ऑल द रेफरेंस पॉइंट व्हाइल डिफाइनिंग अ ट्रांसक्रिप्शन यूनिट इज मेड विद द कोडिंग स्ट्रैंड. ठीक, जो भी हमें रेफरेंस लेना है, ट्रांसक्रिप्शन यूनिट बताना है, हम वो हमेशा कोडिंग स्ट्रैंड के रिस्पेक्ट में बोलते हैं. टू एक्सप्लेन द पॉइंट, अ हाइपोथेटिकल सीक्वेंस फ्रॉम ट्रांसक्रिप्शन यूनिट ये लिया हुआ है. ये हमारा टेम्पलेट स्ट्रैंड है और ये हमारा कोडिंग स्ट्रैंड है. कोडिंग स्ट्रैंड जो है वो 5' 3' बना एंड दिस वाज बेसिकली द RNA. बस अब RNA में क्या है? थाइमीन की जगह यूरासिल है. फाइन. So this is the promoter, this is the transcription side, start side. You have a structural gene. Fine, this is a template strand, this is the coding strand. ये आपको समझ आ ही रहा होगा 5' 3' ही coding क्यों होता है, although it doesn't code for any RNA strand because इसका sequence जो है वो same होता है. Fine, and terminator, here we go. The promoter and terminator flank the structural gene in a transcription unit. ठीक है, क्या करते हैं ये? बेसिकली the promoter is said to be located towards the 5' end upstream of the structural gene. ठीक है, 5' end पे हमने promoter रख लिया. ठीक, the reference is made with respect to the polarity of the coding strand. अब ये आपको समझ आ रहा है polarity किसका लेते हैं हम? coding strand का, जो basically code किसी के लिए नहीं कर रहा होता. It is a DNA sequence that provides binding site for RNA polymerase and it is the presence of a promoter in a transcription unit that also defines the template and coding strands. तो अब जो हमारे पास बेसिकली ट्रांसक्रिप्शन यूनिट है उसी में हमारे पास प्रमोटर है, टर्मिनेटर है और स्ट्रक्चरल जीन है, ठीक है। और ऐसी एक डीएनए सीक्वेंस ही है विच विल प्रोवाइड द बाइंडिंग साइट फॉर आरएनए पॉलीमरेज जहाँ पे बाइंड होगा, फाइन। और इसके एंड इट इज द प्रेजेंस ऑफ अ प्रमोटर इन अ ट्रांसक्रिप्शन यूनिट दैट आल्सो डिफाइंस द टेम्पलेट एंड कोडिंग स्ट्रैंड्स, ठीक। तो बेसिकली प्रमोटर होगा जहाँ से हम स्टार्ट करेंगे ट्रांसक्रिप्शन और वही बताएगा कि कौन सा टेम्पलेट स्ट्रैंड है, कौन सा कोडिंग स्ट्रैंड है। By switching its position with terminator, the definition of coding and template strands could be reversed. ठीक है, तो promoter का अगर आपने change कर लिया polarity, then the game is over. You'll have to change everything. The terminator is located towards the 3' downstream of the coding strand, and it usually defines the end of the process of transcription. Obviously, terminator है तो end को ही define करेगा. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. So then we have the rest of it, which I think we'll be doing in the next lecture probably, because a lot of it in one go might be actually a little disturbing for you also. So keep watching. The second part will be released really soon, and I just hope you could grasp anything out of it. If your basics are done, I think this is a really good technique to study, to learn stuff on the go. So keep watching and see you in my next one. Thank you.